Hi, my name is Sachi Pathak. My study is discussing hydroxysteroid dehydrogenase, or HSD, as a sex-based biomarker in hepatitis C virus-induced cirrhosis, and my mentor is Dr. Rashmi Kaul. Okay, so let's gather some context. HCV is a positive single-stranded RNA envelope virus. Hepatocellular carcinoma, or HCC, is classified as a primary liver malignancy and is a leading cause of cancer around the world. Studies show that chronic HCV and its progression to cirrhosis and cancer development is more prevalent in males as compared to females. Interestingly, pre- and perimenopausal females, along with women on hormone replacement therapy, have been associated with less severe HCV disease. Estrogen receptor signaling is known to regulate inflammation and immunity. So, with these scientific observations, Dr. Kaul's lab has identified that liver estrogen receptors have differential expression at both mRNA and protein levels in both HCV-related cirrhosis liver tissue as well as HCC liver tissue. Moreover, these sex-based differences in receptor expression demonstrates that ER-alpha is basally expressed significantly higher in males than females. With that in mind, we looked at the hydroxysteroid dehydrogenase, or HSD, enzyme family. The enzyme confers steroid hormone synthesis and degradation, as well as cholesterol and fatty acid metabolism. Due to the relationship between HSD and estrogen, and with a strong correlation between HCV-directed cirrhosis and estrogen receptors, the function of HSD enzyme and hormone regulation due to HCV infection demanded more investigation. We hypothesized that chronic HCV infection leads to dysregulated HSDs in male HCV cirrhosis patients, leading to the development of HCC. Male and female de-identified and discarded human liver tissue from normal and HCV cirrhosis liver tissues were used from the NIH Liver Tissue Cell Distribution System at the University of Minnesota. A total of 40 explant liver tissues were used in the study, and each experimental group included 10 tissue samples. So, after explant liver tissue was prepared, we sent them to the Proteomics core lab for processing. The core reduced, alkylated, purified, and digested the proteins. This yielded peptides that needed separation using reverse phase chromatography. Once the peptides were eluted, they were ionized, and finally the core conducted mass spec analysis. So that's a lot of steps, but what did it tell us? 4,443 proteins of different protein families were profiled in normal control and HCV cirrhosis male and female groups. 7 HSD proteins exhibited differential expression. Our research focused on protein HSD17B13 in HCV males compared to HCV females. This differential expression between infected males and females may be induced by HCV infection. We can see the significant relationship here graphically. Okay, so let's put more tangible numbers to this. Only HSD17B13 showed significant sex-based differential expression between male and female HCV cirrhosis groups. HCV cirrhosis males demonstrated significant decrease when compared to HCV cirrhosis females, that is indicated in the third experimental column with the green marking. HSD17B13 showed downregulation in HCV cirrhosis males compared to normal control males, but not significantly. That's the first experimental column with the blue marking. These numbers revealed 3 things. First, HSD17B13 protein levels were significantly downregulated in HCV cirrhosis males when compared to HCV cirrhosis females. 2, HSD17B13 protein may serve as a sex-based biomarker in liver cirrhosis and cancer development. And 3, to the best of our knowledge, this is the first report showing sex-based differences in HSD proteins in pre-malignant HCV-related cirrhosis. From here, we intend to investigate the HSD17B13 protein further to confirm our results. To strengthen our study, including more subjects would help us confirm the significance of our observations. Real-time PCR will help us confirm mRNA expression of this protein in the liver tissues, and IHC studies can help us confirm the presence of the protein. Finally, correlating the clinical data like liver inflammation parameters with HSD protein expression will allow us to understand the role of HSD17B13 in cancer development. Special thank you to my mentor, Dr. Rashmi Kaul, as well as Dr. Subhas, Dr. Anil, and GRA and peer Logan Swope. Thank you for your attention.